Hello everyone and welcome. In this video we're checking out variant 3 of May-June 2024. Before we start, make sure to check out the free notes I have available for you in the comment section down below. They are available for chapters 2 and 1, AS and A2 bio respectively. And also make sure to subscribe to the channel so that you can see what other uploads I'm going to have in the future. Now let's start with question number 1. An eyepiece graticle can be calibrated using a stage micrometer. What is the correct reason why an eyepiece graticle is calibrated? So as he mentioned, the eyepiece graticle will be calibrated using the stage micrometer. Now, if you know what the eyepiece graticle is, well, then you don't need the question here. But if you don't know it, then let me explain it to you. It's supposed to be some what's it called? Some length of divisions that's going to be fitted onto your eyepiece lens. And then you're going to compare it to a set measurement. So let's say that you already know this measurement is 4 micrometers. You're going to compare these so that you figure out the length of one of these guys. And now this length over here is known as the stage micrometer, and one of these guys here is the eyepiece graticle unit. So once you figure out the length of one eyepiece graticle unit in comparison to this length, then you can use the eyepiece graticle right on the eyepiece lens, keep it on the eyepiece lens, and then use it in order to view the different organelles and figure out the sizes and measurements of different organelles. So an eyepiece graticle can be used to make measurements. An eyepiece graticle is magnified by the objective lens. No, as I mentioned, it's fitted onto the eyepiece lens. An eyepiece graticle magnifies the specimen. No, that's the role of the objective and the eyepiece lenses, not the thing that we fit onto the eyepiece lens. An eyepiece graticle makes comparisons. Now, makes comparisons or can be used to make measurements. I believe that the reason we calibrate it is to be able to make measurements. Okay, the image is an electron micrograph of a typical eukaryotic cell. So you have a eukaryotic cell, you have the nucleus. It says, what can be concluded about the eukaryotic cell from the electron micrograph? Okay, so it is an animal cell because it does not have a cell wall. Now if you zoom in here, you can't really do that, but I can. So no, it does not have a cell wall, so it is an animal cell. But okay, it's an animal cell because it has permanent vacuoles. Actually, animal cells do not have a permanent large vacuole like the plant cells. That's not a valid reason. It's a plant cell because it contains chloroplasts. These are most likely, tiny vacuoles like mitochondria, as long as tiny vacuoles like lysosomes and everything. But they don't contain chloroplasts, so no. And because it contains many lysosomes, that's not a valid reason for it to be like a plant cell. I would say it's an animal cell because it does not have a cell wall. Which features are found in typical eukaryotes and also in typical bacteria? Bacteria definitely can respire. I think many people make this mistake of bacteria can't respire because they don't have mitochondria. Well then, they are living organisms, and all living organisms need energy. You move, they move. How do they move if they don't have energy? So they can live. They don't really need mitochondria to live. They have these inner foldings in their plasma membrane which contain respiratory enzymes. So yes, they respire. Messenger RNA binds to ATS ribosomes. Okay. There are no ATS ribosomes in bacterial cells, only 70S. Contain circular DNA? Yes, both of them contain circular DNA, so C. Which type of cell will have the highest proportion of its volume taken up with cell structures bound by a single membrane? So single membrane organelles let's say the endoplasmic reticulum, the Golgi apparatus, the lysosomes, the vacuoles, what are they going to be? So obviously a goblet cell will be a very, high option because goblet cells produce many vesicles which contain mucus, right, by the Golgi apparatus, and so they contain lots of those. Ciliated epithelial cells, I don't think so. Ciliated epithelial cells don't really release anything. They don't really need these vesicles or anything. Okay, red blood cells. Red blood cells do not have a bunch of single membranous organelles. And companion cells, same situation. I think goblet cells definitely going to be your number one answer always. Okay, what causes the phosphate heads of phospholipids to become polar? Phosphate heads are actually negatively charged, and so they are polar. And the phosphate heads are joined to water molecules by hydrogen bonds. The phosphate heads are insoluble in water. The phosphate heads become ionized in water. This is your answer over here. Now, as a result of phosphate heads being polar, they can form hydrogen bonds. It's not the reason they are polar. I hope this is clear to you. Again, if something is polar, it can make hydrogen bonds. If it's not polar, then it can't make hydrogen bonds. If something can make hydrogen bonds, then it's polar. But just because something is it's just because it can form hydrogen bonds, it does not mean that this is the reason it's polar. The reason it's polar is because it has this difference in charge. Okay, so and this uneven distribution of charge, or the fact that it forms ions, like the phosphate groups, which are basically phosphate ions. So definitely C. Okay, which statements describe features of cellulose adapted for its function in plant cells? Okay, So cellulose and adaptations. Now, cellulose molecules, there are 3 cellulose molecules coil around each other to form a triple helix structure. This is collagen, not cellulose. Many hydrogen bonds form between adjacent cellulose molecules. That is actually true, and this allows cellulose molecules to form what's it called, microfibers, which then join together by more hydrogen bonds to form cellulose fibers, which are really, really high in tensile strength. And then covalent bonds form between adjacent cellulose molecules. Okay, no, covalent bonds do not form. Hydrogen bonds do form. Hydrogen bonds are intermolecular forces of attraction, not covalent bonds. Covalent bonds are really strong and hard to break. Hydrogen bonds are like really weak and easy to break, so that's wrong. So it will be 2 only D. Which structure shows alpha glucose? Okay, which one is alpha glucose? Pretty easy to identify this one. You just go down. This is not down. So look at the hydroxyl groups and go down, down, up, down. B. Okay. Now, what else do we have here? We have what cannot occur as a result of a condensation reaction, breaking of a glycosidic bond. Okay, condensation reactions mean the formation of a bond via removal of water. So it's not going to be breaking down of anything, it's going to be the formation of things. Formation of a disaccharide can occur, joining together can occur, production of something can occur, however, breaking cannot occur. Okay, which fact about the quaternary structure of proteins is correct? Okay, quaternary structures just means that it's more than one polypeptide chain. So it consists of 4 polypeptides. That's not necessary. Okay, so that's not true. Collagen has 3 polypeptide chains. It's not. It also has a quaternary structure. Depends on the presence of metal ions. No, not at all. Okay, I think many people confuse quaternary structure with hemoglobin structure, and so this may be a tough question for them. But again, hemoglobin has quaternary structure because it has more than one polypeptide chain. It's not that it has more than one polypeptide chain. It just has 4, and the specific structure of hemoglobin is the quaternary structure. That's wrong. Okay, again, quaternary structure just means to have more than one polypeptide chain. Depends on the primary structure of polypeptides. It's made of alpha and beta polypeptides. Okay, not necessarily. Yes, all structures depend on the primary structure. Okay. The diagram shows different molecules in a solution. So which statements could explain what happens when some of the molecules are mixed together? So molecule P forms an enzyme substrate complex with a non-competitive inhibitor and molecule Q. Okay. So firstly, molecule Q does not look anything like an non-competitive inhibitor because let's just say that it's perfectly complementary to the active site of the enzyme. Usually if you have a non-competitive inhibitor, it'll look actually more like it's something that can bind to the other site, the allosteric site of the enzyme, not directly to the active site, because if it can't bind to the active site, then it's a competitive inhibitor. Molecule Q binds to molecule P, increasing the activation energy. Okay, if Q was the substrate and P is the enzyme, then I have to say that the enzymes decrease activation energy, not increase them. And then molecules R and S bind to the active site of molecule P. That definitely could work. It could be like a condensation reaction where this guy from here and these guys from here are going to bind to this enzyme. So you could see how this works. Something like this, and then they join together. So yes, this is true. And the molecules S and R are the products of the breakdown of molecule P. I don't think so. I think P is the enzyme. Okay. Moving on, we have the effect of substrate concentration of an enzyme catalyzed reaction was measured in 3 different conditions: without an inhibitor, with a competitive inhibitor, with a non-competitive inhibitor. Immediately looking at this graph, you can figure out which one is which. You could see the non-competitive inhibitor is definitely number 2 because you could see the Vmax in the end is going to be reached. However, the Km value is increased. It's just that the affinity of the enzyme to the substrate decreases. Meanwhile, here, what happens is that the Vmax actually decreases. However, the Km remains the same. So what exactly do I mean by Km? What do I mean by Vmax? Let me explain to you really quickly. The affinity of an enzyme to the substrate means the ability of the enzyme to quickly match and find the substrate. Okay. So when you add a competitive inhibitor, which basically looks exactly like the substrate, it's going to distract the enzyme and not allow it to find and match with its substrate directly, and so that decreases the affinity of the enzyme to the substrate. Therefore, we need a higher substrate concentration for the enzyme to be able to spot it and see it directly without being distracted by the competitive inhibitor. Which means that the substrate concentration being higher means that the Km increases, because the Km is supposed to be the substrate concentration at which the enzyme reaches half its Vmax. Now, if you don't know what this definition is, I would really encourage that you memorize it. However, for now, let me explain to you what it is. Vmax is the maximum rate of reaction, which I mentioned before, right? So half the Vmax would be half the maximum rate of reaction. Now, the greater the affinity, the lower the substrate concentration required to reach this half the rate of maximum rate of reaction. But when we add a non-competitive inhibitor, like as you can see in number 2, what happens is that the Km, the substrate concentration at which half the Vmax is reached, is definitely bigger, right? It's bigger than this value here. And therefore, what's it called? This means that the enzyme affinity to substrate has been decreased. Okay, now moving on to the next one, you can clearly see that the Vmax here is lower, which means it's a non-competitive inhibitor, because non-competitive inhibitors, they work to destroy the enzyme's like shape and active site and tertiary structure, and so overall they just decrease its rate of reaction, decrease the amount of enzymes that actually work, and therefore there are other limiting factors affecting the rate of reaction other than the substrate concentration, so you can see we don't end up reaching the same rate of reaction. Okay, which row is correct? Without an inhibitor is definitely 1, with a competitive inhibitor is 2, with non-competitive inhibitor is 3. Which aspect of enzyme activity can be compared by Michaelis-Menten constant? Okay, so as we mentioned, Michaelis-Menten constant is the Km value, which we mentioned was the substrate concentration at which we reach half the maximum rate of reaction. So it's just that we use it in order to compare the affinities of the enzyme to their substrate. So affinity of an enzyme to at different substrate concentrations, the activation energy of the enzyme, affinity of different enzymes for their substrate, and then yes, so I would say affinity of different enzymes to their substrate, because the affinity remains the same no matter the substrate concentration, unless obviously you add something like a competitive inhibitor. The number of substrate molecules on one enzyme molecule can convert to a product in a second, and it's called the turnover number. This number is obtained when all conditions are optimum for the specific enzyme catalyzed reaction. So you can see that it's going to be a higher turnover for things like catalase, carbonic anhydrase, which means basically that they are, they have a greater rate of reaction compared to that. How many times faster at converting substances to products is catalase compared to phosphatase? So we go to look at the turnover rate here and turnover rate here, and you divide them by each other. This way you'll figure out how many times faster they are. If you divide, let's go ahead and do this, and you're going to get, well, let me just check, it's 1, 2, 3, 4, 5 zeros exactly, so it'll be 2884 approximately. That will be C. Okay, a red indicator solution was mixed with agar, and resulting solid was cut into small cuboid blocks, and the blocks were placed into an acid which turns yellow. It turns the indicator yellow, and all other variables were kept constant. The dimensions of the 3 blocks are used are shown. So you have 3 by 3 by 3, 8 by 8, and then 11 by 11 by 11. So obviously the smallest block is going to have the biggest surface area to volume ratio. So which row shows the correct surface area to volume ratio and the time taken for the block to turn yellow? So remember, with the greater surface area to volume ratio, the greater the rate of diffusion, and therefore the smaller time it takes for the things to diffuse. And so therefore we should expect the shortest time for block 1, the longest time for block 3, and the middle for block 2. So the surface area to volume ratio should be greatest for block 1, so we expect this is definitely low, and then the time should be short. Okay, for surface area to volume ratio of the second one, it should be lower, and then the time should be longer, and lastly it should have the lowest and the longest time. Now you may want to check the second one, and you would see that actually the times are wrong. You may want to check the first one, and you see that the ratios are wrong, and that's why we don't really need to calculate using the calculator and figure out the surface area to volume ratio of each one of them. Okay. 15. The statements describe some events in the process of exocytosis of a glycoprotein molecule. Okay, membrane of the Golgi body folds around the glycoprotein molecules. Vesicles bound to and fuse with cell surface membrane. Vesicle attaches to the microtubules through the cytoplasm. Secretory vesicles form. Okay. So what's the correct order of exocytosis? First things first is what's it called, the Golgi body is going to be like, membrane of the Golgi body folds around the glycoprotein molecules. And then it's called vesicles bind to and fuse with the cell surface membrane. Vesicles attached to the microtubules through the cytoplasm, and then secretory vesicles form. I think it's going to be 1, then 4, and then what's it called, 3, and then 2. Okay, what do I mean by this? Firstly, what happens is the glycoprotein molecules are going to be formed in the Golgi body, and they're going to be released in vesicles from the Golgi body, and then these vesicles over here are going to be formed, and then what's it called, 3 is basically when vesicles start to move across the cytoskeleton of the cytoplasm, and 2 is going to be when they're released via exocytosis. So it's 1, 4, 3, 2. Okay, 4 cylinders were identical in size, A, B, C, D, were cut from potatoes that had been stored for different lengths of time. The cylinders were weighed, immersed in 10% salt solution, 45 minutes, and then reweighed. And the percent changes in mass was then calculated. Which cylinder had water potential similar to the 10% salt solution? So the one that has the least change in mass is going to have the closest water potential to the what's called salt solution. And so we look at these numbers and we see that minus 0.9 isn't as big as minus 7.2 or plus 3.4, and therefore that means it has the closest water potential, the closest concentrations as the salt solution, and therefore not much osmosis or diffusion happened in that potato. Okay, the contents of daughter cells are compared to the parent cell after one cell cycle. Which row is correct? Okay, number of chromosomes. Now the daughter cell over here is going to have the same number of chromosomes to the parent cell, because again it's mitosis, we produce genetically identical cells. So if it's genetically identical, we should have the same number of chromosomes. Volume of cytoplasm, however, is going to be a little bit smaller because the daughter cell is going to be formed by the division of this mother cell. And therefore, since it divides, it's going to turn into 2 smaller cells. So it should decrease a little bit. Length of telomeres. Now, the length of telomeres after every time DNA replication happens, which is basically just before the cell cycle happens, that when mitosis happens, that's going to decrease the length a little bit, especially if it's not a stem cell. The length of the telomeres should decrease. So the answer would be D. Moving on to 18, a high power photomicrograph shows a cell in a stage of mitosis. The chromosomes are visible and lined up along the cell equator, but there is no nuclear envelope. Which stage of mitosis is shown by the photomicrograph? Okay, so there's no nuclear envelope and the chromosomes line up along the equator. It's pretty obvious that it's metaphase. Remember, metaphase stands for middle. And then which statements about the cell cycle are correct? The cell cycle includes interphase and mitosis. Okay, the cell cycle is going to include all the processes, whether it's interphase, mitosis, S phase. Interphase is going to include G1, S phase, and G2. And then DNA replication takes place in interphase. That is true. And then a cell can remain in interphase for several months. That's actually correct. So it's really 1, 2, and 3. Okay, scientists stain the chromosomes of plants with a fluorescent dye to observe the telomeres. This cell has 38 chromosomes. How many telomeres will the scientists observe in one nuclei during the telophase of mitosis? Okay, in one of the nuclei. Now, each one of these nuclei, they're going to have what's called this set of chromosomes, right? So it's not going to be containing it's not going to contain let's say twice the number of chromosomes. It's not going to contain half the number of chromosomes. It's not like a gamete, so it should have all 38 chromosomes, right? But the 38 chromosomes are not going to be this shape. This is only going to be during mitosis, during prophase, just after S phase happened, synthesis happened, and there was DNA replication. But now, after anaphase happened and the 2 sister chromatids were split apart, each nucleus is going to have the 38 chromosomes with only 1 chromatid, and each chromatid is going to have only 2 telomeres, and therefore just multiply this number by 2, which will give you 76. Okay, observative replication of DNA. The double helix is unwound by an enzyme, which diagram shows how the strands are copied. So you have to remember that the strands are going to be copied. When DNA polymerase copies the strands, it goes in the 5' to 3' direction. So we're going to look for DNA, and this will be the leading strand, which means basically that when DNA polymerase copies that strand, we're not going to have any gaps or Okazaki fragments. We're going to have a complete line, and everything's going to be fine. So we need the 5' to 3' strand to be our leading strand, and so that you can see this is straight and there are no gaps. Meanwhile, the 3' to 5' strand is going to be the lagging strand, where then when DNA polymerase goes, it's going to go in the opposite direction, and it's going to end up forming these fragments, these Okazaki fragments. Now here, the difference between D and C, we eliminated A and B because they started with 3' and they went in the 3' to 5' direction. And so the DNA polymerase does not work that way. It should go from the 5' to 3', and this should be the leading strand. Now here he's labeled it as lagging strand, so this is wrong, and therefore the answer should be D. Okay, a transcription error results in a deletion of one nucleotide from the middle of a primary transcript. Okay, so there's a transcription error where we have the primary transcript of mRNA, and it's what happens is one nucleotide from the middle was actually removed. Now, which statement describes one possible effect of this deletion on the protein translated from this mRNA? So this protein could have a missing amino acid, right? So the protein would be unchanged, as the amino acids can be coded by another codon, formed by after deletion. I think that when you remove one nucleotide, this changes, like this removes like one one letter, and when you remove one letter, this could cause like the whole thing to shift. So it's called a mutation caused by a shift, or it could be that there's like a deletion, which like it's just at the end maybe or something. So but since it's just mentioned in the middle, I feel like it will cause the whole thing to shift, and so it may change a bunch of amino acids. So the tertiary structure of the protein is not affected as only one amino acid has been changed by the deletion. The protein will be unchanged as the same amino acids can be coded by another codon formed by the deletion. The sequencing of amino acids will be different as the codons from the deletion onwards are changed. This is exactly what I'm talking about. Which statements correctly describe the process of translation? The process of translation, the nucleotide sequence on the mRNA molecule is used to produce a specific amino acid chain. Okay, so mRNA is going to bind to the ribosomes, and what happens there is that the tRNA is going to come in and with like the complementary anticodon carrying its amino acid, and then this amino acid is going to bind to the amino acid, the polypeptide chain that's being formed. So the nucleotide sequence mRNA is used to produce a specific amino acid chain. That is kind of true. Okay, and then a section of DNA is copied into an mRNA molecule by RNA polymerase. No, this is transcription. We're not talking about transcription. Okay, a polypeptide is produced because anticodons on tRNA molecules attach to mRNA codons through peptide bonds. Definitely not through peptide bonds, but like by brief hydrogen bonds that are quickly formed between the two. So, no, it's just one really. Okay. A molecule of mRNA was used in translation. Part of its sequence is shown. You can see it's GAU, CUG, UAA, CGG. There were no introns present in the section of DNA to transcribe to make this mRNA. So you have no introns. Okay, so that means it's just all, coding parts. It says, what is a sequence of non-transcribed DNA strand for this section? So again, non-transcribed DNA strand. So this is what they're talking about. They're talking about the non-transcribed strand. Alright, so remember, there is the coding strand, and then there is just like the non-transcribed strand of DNA. So what happens is that during transcription, helicase is going to unzip the DNA, and the coding strand is going to be the one copied into complementary mRNA. And so when it's copied into mRNA, okay, it's going to be, obviously, since it's complementary to this strand, it's also going to be the same as her because she is also complementary to this strand. So that means mRNA is pretty much the same as the non-transcribed strand, except for every U will be a T because it's DNA instead of mRNA. So it should be GAT, CUT, and then TAA and CGG. So yeah, that's right. Okay, which properties of water are dependent on hydrogen bonding between water molecules? Is it cohesion, high latent heat evaporization, solvent action, high specific heat capacity? Now, to some extent, all of these guys actually, they are all dependent on hydrogen bonding. I mean, cohesion is supposed to be the bonds that are formed between water molecules and how they're able to hold each other together. And so that these kinds of bonds are hydrogen bonds. And then high latent heat evaporization, really, it's really high because we need to break these hydrogen bonds, and there's so many of them. And so it takes us so much energy to break all of them down. And solvent action, because it's able to form hydrogen bonds, it's able to dissolve all of these things because it's like polar, and so it's able to dissolve other polar molecules. And then high specific heat capacity, it's also going to be dependent in the same way that high latent heat of vaporization is dependent. But somehow in this question they want you to eliminate one of them. So which one do you guys think is the least one that is dependent on hydrogen bonding? Yes, they're all dependent, but in some way or another, one of them is not that dependent. So in this case, if we eliminate cohesion, that is the biggest mistake that you can do. I mean, come on, cohesion is basically hydrogen bonding between water molecules. So we need to keep cohesion with us, okay? But then moving on to high latent heat vaporization, if you eliminate it, then you have to also eliminate high specific heat capacity. So you can't really eliminate high latent heat vaporization or high specific heat capacity. So you can only really eliminate solvent action, which means it's B. And if you think about it, really, it's not because water can form hydrogen bonds. It is actually able to be a solvent, because not all molecules that it's going to be dissolving are going to be able to form hydrogen bonds with it. Some things like, let's say, ions, they are going to form this shell about the ions. However, they're not going to form hydrogen bonds with them. So yes, I guess you could say this. Okay, which substances in xylem tissue are impermeable to water and prevent the collapse of vessels? Okay, impermeable to water, prevent the collapse of vessels. Impermeable to water is going to be lignin, definitely not cellulose. Prevent the collapse of vessels are going to be both cellulose and lignin. Okay, so it would be The electron micrograph shows a longitudinal section through the phloem tissue, with students' drawings of sieve tube element is correctly drawn and labeled. Okay then, so we have the cell wall, the sieve plate, and then we have the mitochondrion and the sieve plate. Interesting, because like it's such a bad drawing of mitochondrion. And it's not really obvious it's like mitochondrion or anything. It's like these black drops. So I'm not really sure if these are like mitochondrion actually or not. And then cell wall, cytoplasm, and sieve plate. I think that the only thing that you can actually label here would be the cell wall and the sieve plate. Maybe even the cytoplasm, you could see like the edges. So yeah, I would definitely choose C. And no, I would never choose D because D is colored and you shouldn't be coloring your drawings. Not here, not in the lab. Okay. Carrier proteins in the cell surface membranes of companion cells are involved in the transfer of assimilates to phloem sieve tubes. The diagram represents the use of two types of carrier proteins in this process. So you have carrier proteins, you have the companion cell, and you could see what's it called, the two types of carrier proteins. So there's a pump, and then there's a co-transporter. Okay, so what are the substances labeled X and Y? Okay, then X over here, hydrogen ions and sucrose, or hydrogen ions and glucose, or what? Okay, so since we're talking about the transport of so you could see here how there's X and then it's changed to X and then again X to Y. Now sucrose is only going to ever be co-transported. However, hydrogen ions are going to be pumped, are going to be pumped against the concentration gradient into the what's it called cell membrane and cell wall of the neighboring cell of the what's it called companion of the neighboring cell of the companion cell. And then what happens then is that what's it called, these guys over here are going to diffuse back, but when they diffuse they're going to go through special co-transporter proteins and carry with them the sucrose. So yes, hydrogen ions, and then Y should be sucrose. Okay. The diagram shows pressure changes during 2 cardiac cycles. Which arrow indicates atrial systole? Okay. Which one indicates atrial systole? So A is going to be ventricular systole. We're not going to be choosing that. And then C or B. Okay. Well, obviously it's not going to be C. C will be like the end of atrial systole. Like that's going to be like, okay, I'm done. Like now we're gonna close the atrioventricular valves because now there'll be like the what's it called, ventricular systole. So it should be B. Okay. An irregular heartbeat may be the result of ineffective electrical stimulation of the artery. Which area of the heart could be damaged, causing this irregular heartbeat? Irregular heartbeat. So an irregular heartbeat may be the result of ineffective electrical stimulation. So it's called irregular heartbeat is going to be caused by this atrial node. It's going to be actually what controls the rhythm. It's known as the rhythm maker. So when it's like there's an irregular heartbeat, it should be because the atrial node is not working properly. Shows the effect of 3 different concentrations of carbon dioxide on the oxygen dissociation curve for human hemoglobin. You have the percentage saturation of hemoglobin with oxygen, and then you have which effect does increasing carbon dioxide concentration have on hemoglobin. Okay. When you increase carbon dioxide concentration, you decrease the affinity of hemoglobin to oxygen, and therefore you decrease the saturation of hemoglobin with oxygen. And so you can see actually, it makes hemoglobin less efficient at taking up oxygen, less efficient at releasing oxygen. No, obviously less efficient at taking up oxygen, more efficient at releasing it, because it will want to bind to more carbon dioxide. Now, which reactions take place in the blood and the capillary next to an alveolus? Next to an alveolus is going to be where carbon dioxide will be released, and oxygen will be attaching to hemoglobin. So yes, oxyhemoglobin will form, and then will carbonic acid be formed? Actually no, carbonic acid will be broken down, right, and it forms carbon dioxide so that carbon dioxide can leave and go into the lungs. And then you have carbonic acid again for the dissociation. We have hydrogen carbonate ions, and they bind to the hydrogen ions from the hemoglobinic acid, which dissociated as well, and then they're going to form hydrogen carbonate. So they're going to form carbonic acid. Carbonic acid will dissociate into carbon dioxide and water. Carbon dioxide is going to be released into the lungs. Oxygen from the lungs will bind to hemoglobin, forming oxyhemoglobin. So it should be just 1. So that's B. Okay, what structure of the gas exchange system will always contain cartilage? Always contain cartilage will be definitely not the capillary, definitely not the bronchiole or the alveoli. It will definitely always be the bronchus. Okay. Exchange of carbon dioxide and oxygen occurs between air in the alveoli and blood in the capillaries of the lung. Which partial pressures of the gases will allow gases exchange to occur? CO2 in alveolar air, CO2 in the blood, oxygen in alveolar air, oxygen in the blood. Okay, so CO2 and alveolar, which partial pressures will allow the gases exchange in the lungs? So we should have a, What's it called? We should have a low partial pressure of carbon dioxide in the lungs, and then what's it called? What exactly we're going to have like a now. Obviously, in the alveolar air, we should be having a low partial pressure of carbon dioxide, but in the blood, we should be having a high one because we're trying to get rid of CO2 in the blood. However, in the blood, we should have low oxygen, and in the air, we should have really high oxygen. So, yes, 13.9, I guess, and then maybe 5.3, and 6, and 5.3. So this is going to be it. Now, the plan diagram shows a cross-section of trachea, which labeled tissue prevents the trachea from collapsing. So we need the cartilage rings. These are going to be preventing the trachea from collapsing, and that's going to be, well, A. Okay. Which layers of cells does an oxygen molecule diffuse through when moving from an alveolus into an alveolar capillary? From an alveolus into an alveolar capillary. Okay, so you have your nice looking let me try my best here, guys. Okay, so you have the alveolus, and then what's it called? You have the alveolar capillaries. So if you zoom in, there are a bunch of capillaries out in the alveolus. Now, in the alveolar capillaries, is saying which layers of cells does an oxygen diffuse through? In an alveolar capillary, you have to know that it's only going to contain this endothelium. Yes, endothelial cells are going to be the ones oxygen is going to contain. I don't know why I did the drawing. I feel like it's useless now. Okay, so what's it called? Next we have alveolus, obviously squamous epithelium. Moving on from here would be bacteria, maybe classified according to differences in cell wall structure. The differences are shown in the gram stain, and you could see that the diagram shows part of a gram-positive bacteria and part of a gram-negative bacteria drawn to the same scale. So the external environment, the gram-positive bacteria, the peptidoglycan layer, the periplasm, which is a watery fluid containing proteins, and then we have what's called cell surface membrane. Moving on from here will be a cytoplasm of a gram-negative cell wall of a gram-negative bacteria will contain an outer layer of carbohydrate and lipid, so not of peptidoglycan. And then what's called periplasm, which we mentioned was the watery fluid containing proteins. A peptidoglycan layer comes next, and then again more layers of water fluid containing proteins, and then cell surface membrane. So he says here that the antibiotic penicillin kills bacteria by inhibiting the synthesis of cell walls during bacterial cell growth. Which type of bacteria will be killed by penicillin more easily, and why? Okay, so remember, penicillin is going to inhibit the formation of cell wall. Which one is going to be killed more easily? So let's see. Gram-positive bacteria, because the peptidoglycan layer is exposed to penicillin directly. The peptidoglycan layer is exposed to penicillin directly, or the gram-positive bacteria because it has a thinner layer surrounding cell membrane. Overall, the gram-negative bacteria because it has a thin peptidoglycan layer can be broken down faster. Gram-negative bacteria because it has a more periplasm available, which gives a weaker structure. Okay. Nothing to do with periplasm because we never discussed periplasm before. However, what I would have to say is, how does penicillin work? Do you guys remember how penicillin works? So how it works is basically it will bind to the cell wall, and then when it binds to the cell wall, it's going to inhibit the formation of the cross-links and everything, and so it's going to stop the formation of the cell wall and everything. So if you guys remember, do you guys remember the slimy capsule or anything? Now, these guys were supposed to be there in order to increase the what's it called antibacterial, being able to protect themselves. So it's defense mechanism and being more antibacterial. So no, what did I just say? Being more able to fight off antibiotics. So what's it called? When it comes to just having a peptidoglycan layer, like we mentioned in option A, you're just directly exposed to the penicillin. Whereas if you had something like an outer membrane with carbohydrates and lipids and this capsule that can protect the cell wall, then in this case you're definitely more able to protect yourself against the effect of penicillin. So gram-positive bacteria, because what's it called, penicillin is exposed to it's exposed to penicillin directly. Okay, which facts relate to the fact that TB or its pathogen? So viruses change their antigens to a limited extent. TB is caused by only one species of pathogens. HIV or AIDS makes the bacterial infection worse. The pathogen may be transmitted by ingestion. The pathogen may be transmitted from animals. Multi-drug resistance occurs. Okay, firstly, but TB is a bacterial, it's not going to be caused by viruses, so we can just cancel out 1. TB is not caused only by one species of pathogen. It could be caused by 1 or 2. Mycobacterium bovis, Mycobacterium tuberculosis. HIV or AIDS makes the bacterial infection worse. HIV or AIDS does make the infection worse because HIV and AIDS may decrease the immune system's ability to function, and TB likes to hide from the immune system until it's at its weakest point, so then it starts to replicate and take over the body. So yes, okay. And then the pathogen may be transmitted by ingestion. Yes, if you eat things like raw meat or something, that's going to cause you can get the pathogen. And then pathogen may be transmitted from animals. That is also true. It's transmitted by animals, from cattle, from sheep, from all these things. Multi-drug resistance occurs for all bacterial infections. You expect multi-drug resistance. So 3, 4, 5, and 6. D. Which events listed occur during the primary immune response to a specific pathogen? So the activation of B lymphocytes to produce plasma cells and what's it called memory cells, phagocytosis of invading pathogens, and macrophages by macrophages. So that's going to be number 1 always. Okay, phagocytosis is going to happen first, and then T helper cell activation, production of T killer cells, which come in maybe like later on. Let's see the 4th option, expression of antigens on phagocyte cell surface. That is right. So the macrophage is going to turn into the antigen presenting cell. It's step number 2. And then T helper cell activation and everything. But let's continue. 5, production and release of antibodies. That's going to be actually the last step. So let's get this delayed. Let's get back to this later. As well as like production of memory cells should be one of the last steps. But obviously, because one says activation of B lymphocyte to produce plasma cells and memory cells. Now, since you're activating it to produce plasma cells are the ones that are going to produce and release your antibodies, and therefore it's going to be 1, then 5. So I would have to say, yes, T helper cells are going to be number 3, and then this will be 4, and this will be 5. So it's going to be 1, 2, 3. Okay, it's going to be 2, 4, 3, 5, 1. B. Okay. Influenza is an infectious disease caused by a virus. It is possible to have influenza more than once. Which statements explain why it's possible to have influenza more than once? Okay, so the viral antigens change as a result of mutations. This is true. Mutations can cause the disease to have different antigens. So you're gonna have to make totally different antibodies and everything. So you're going to need that's going to be number 1. The immune system may be weak and make few baby memory cells. I guess so. If you make few baby memory cells, then this will not be enough. Yeah, so it's kind of like a suggestion. If the immune system is weak and it only makes a few B memory cells, and the B memory cells as I mentioned, if the immune system is weak and it can't get activated and produce plasma cells, which then don't produce antibodies, I think this is another option. Okay, untreated HIV has resulted in lower T helper cell count. Of course, I mean, if it's untreated HIV, then you totally expect someone to have influenza maybe 500 times because they have 0 immunity. So 1, 2, and 3. So that'd be A, and this will be the end of today's video. So I hope this video was useful for you guys. If it was, make sure to like and subscribe, and I hope you guys like this video. Anyway, I'll see you guys in the next one. Bye.